All right, so today we're playing detective. But, our crime scene is a petri dish, and the culprits, they're completely invisible. We're talking about microbial forensics, the whole process of how you actually culture and identify the bacteria that make us sick. So, let's get right into it. I mean, this is the big question, right? How do you identify an enemy you literally can't see? When a patient is sick, a scientist gets a sample, and it might look like, well, nothing. So, how do they go from that to pinpointing the exact microscopic troublemaker causing all the problems? It's probably the most critical first step there is in fighting an infection. And here's the blueprint for our entire investigation. You can see it's not just random guesswork, it's a really systematic procedure that microbiologists follow. First, you get the sample, you might do an initial stain, then you put it on the culture media, and then you just have to wait. After that, you'll see the path kind of splits based on what starts to grow. This is our roadmap for the whole process. All right, so just like any good investigation, it all starts with preparation. Our first move is setting up the lab. We got to choose the right tools for the job because we have to be absolutely sure the evidence we're gathering is clean and totally reliable. Okay, so first up, we have to choose our evidence board, which is really the culture media we're going to use. Think of blood agar as casting a really wide net. It grows pretty much everything, both gram-positive and gram-negative bacteria. But then you have something like McConkey agar, which is way more selective. It's more like a targeted search. It only lets gram-negative bacteria grow. Using both helps us narrow down our list of suspects right from the very beginning. Now this is the golden rule of microbiology, and it is absolutely non-negotiable. Aseptic technique at all times. Seriously, think of it like a forensics team taping off a crime scene. Any little bit of contamination from outside bacteria, we're talking from the air, from your hands, from anything, it can completely ruin the evidence and point you to the wrong culprit. So how do you actually do that? How do you keep everything sterile? Well, these are the rules of evidence, so to speak. You keep your culture plates closed as much as humanly possible. You actually pass the neck of your test tubes through a flame. This creates an updraft of hot air that pushes airborne germs away. You even hold the tube caps with your pinky finger so you never have to set them down on a potentially contaminated surface. And that little wire loop you use for streaking, you flame it until it's right hot before and after every single use. Every single one of these steps is deliberate and crucial. Okay, so our lab is prepped, our tools are sterile. Now we get to what is maybe the most critical skill in this whole process. We need to isolate a single suspect from a crowd of literally millions. The goal here is crystal clear. See, the original sample is a jumbled mix of all kinds of different bacteria. To figure out who our pathogen is, we have to separate it from all the background noise. What we're aiming for is a pure culture. That means a colony that we know grew from just one single bacterial cell. So how do you do it? It's a technique called the quadrant streak method. You start by taking your sample and streaking it pretty heavily in one corner of the plate. We'll call that quadrant A. Then, and this is the most important part, you flame your loop to sterilize it completely. Then you drag just a little bit from A into quadrant B, thinning it out. Flame the loop again. Drag from B into C. Flame it one last time, and then drag from C into the final quadrant, D. With each step, you're just diluting and diluting the sample. And you can see the result perfectly right here. You've got that super heavy growth back in quadrant A where we started. But by the time you get all the way over to quadrant D, the bacteria are so spread out that individual cells finally have enough room to grow into their own little distinct, isolated colonies. And boom, we've separated our suspects. All right, our plate is streaked, the trap is set. Now comes the part of the investigation that's all about patience. It's the stakeout. We have to give our suspects just the right conditions and, well, time for them to reveal themselves. So how long do we wait? Well, for most of the usual suspects, this is the magic number. We stick the plates in an incubator and give them about 18 to 24 hours. That's usually all it takes for visible colonies to pop up. But what temperature do we need? Well, it really depends on the suspect's usual hangout spot. If you're hunting for a pathogen that likes to invade our internal organs, you've got to mimic human body temperature, so that's 37 degrees Celsius. But for bacteria that live on the skin or just out in the environment, you're going to want to set that thermostat a little bit lower. Okay, but what about the really picky, kind of high-maintenance pathogens? Some of them, called capnophiles, need a lot more carbon dioxide in the air to grow. And for that, there's this amazingly clever low-tech solution called the candle jar. You literally just put your plates in a big jar, light a small candle inside, and seal the lid. Before the flame goes out, it uses up some of the oxygen and pumps out CO2, creating that perfect CO2-rich atmosphere they need. It's brilliant. All right, the stakeout is officially over. We come back to the lab 24 hours later, we open that incubator, and here it is, the moment of truth. What was once invisible has now become visible. It's time to analyze the clues.
You see, every bacterial species grows into a colony that has its own unique set of characteristics. You can think of these as the bacterium's fingerprints, and we look at everything. How big are the colonies? What color are they? What's their overall shape? How raised up are they from the agar? What do the edges look like? What's the texture? And yep, believe it or not, even what they smell like.
And this is the beautiful and incredibly precise visual language that microbiologists use. A colony isn't just round. I mean, is it circular with a nice smooth edge? Or is it rhizoid with these weird root-like branches spreading out? Is it flat against the surface, or is it convex, popping up like a little dome? Every single one of these details is another clue to its true identity. And here you can see it in action on some real culture plates. You can start to identify different suspects just based on these visual clues. On this particular type of media, E. coli often shows up as these distinctive red colonies. Proteus, on the other hand, is famous for this swarming growth that just takes over the entire plate. And then you have Streptococcus, which makes these teeny tiny little pinpoint colonies. It's pure pattern recognition, and it's awesome. So just by carefully observing all these colony characteristics, we can make what's called a presumptive identification. Now, this isn't the final 100% confirmed ID, but it's a really strong, evidence-based, educated guess. And this is so important because it allows a doctor or a vet to get a head start and begin a targeted treatment while they wait for the final conclusive results to come in. So there you have it. We started with a completely invisible smear. And by following this systematic process of culturing, isolating, and observing, we've turned it into a lineup of visible, distinct suspects. This whole process gives us that crucial first answer. But the investigation isn't totally over yet. What's that final step to get a 100% positive ID? Well, that's where the next phase, things like biochemical testing, comes in to confirm the verdict and finally close the case.